Hello, everyone. Before we begin today's lesson, I would like you to ponder a question. How do the vegetables we consume daily grow from a tiny seed into a lush plant? In addition to traditional sowing methods, modern technology offers us another efficient and remarkable propagation method, vegetable tissue culture seedling. This technique not only enables the rapid propagation of a large number of vegetable plants, but also preserves the desirable characteristics of the varieties. Through studying vegetable tissue culture seedling, we aim to understand the fundamentals and key aspects of tissue culture technology and master its application in vegetable seedling production. Today's lesson consists of 4 main topics. 1. Basics of tissue culture technology. 2. Key steps in tissue culture technology. 3. Application of tissue culture technology in vegetable seedling. 4. Operational examples and case analysis. Let's begin with the basics of tissue culture technology. Tissue culture is a technique that uses plant cells, tissues, or organs for ex vitro cultivation under sterile conditions. By providing suitable nutrients and hormones, it induces differentiation to form new plants. The principle of totipotency in plant cells indicates that each cell of a plant carries all the genetic information required to form a complete plant, capable of developing into a full individual under appropriate conditions. Tissue culture technology has significant application value in rapid propagation, variety improvement, virus-free cultivation, and genetic engineering. The development of tissue culture technology has evolved from early explorations and practices to technological innovations and expanded applications, ultimately leading to contemporary development trends. Strict laboratory conditions are essential for tissue culture technology. First, a sterile operating environment is necessary. The laboratory must be equipped with sterile workstations and disinfection equipment to ensure that the cultivation process is free from microbial contamination. Second, culture equipment and materials are required, including culture medium preparation devices, illuminated incubators, and temperature control devices to provide the appropriate physical conditions for plant tissue culture. Lastly, the qualifications of technical personnel are crucial. Operations and management should be conducted by professionals with relevant backgrounds in biology and botany. Now, let's delve deeper into the key steps in tissue culture technology. You must understand the selection and preparation of culture media, including the components, types of media, and sterilization and storage of media. For example, culture media typically contain inorganic salts, organic compounds, and hormones, and must be adjusted according to the purpose of cultivation and the species of plants. Additionally, common types of culture media include MS medium, B5 medium, each with its characteristics and suitable applications. Moreover, culture media need to be sterilized using high-pressure steam to ensure sterility and stored under suitable conditions. The commonly used conditions for high-pressure steam sterilization are 121.3 degrees Celsius for a duration of 15 to 30 minutes. For tissue culture, the selection and handling of explants are especially important, encompassing type, disinfection, and inoculation. Types of explants include shoot tips, leaves, and embryos, each with different cultivation potentials. We can use disinfectants like alcohol or sodium hypochlorite to disinfect the surface of the explants, removing potential contaminants. During inoculation, it is vital to inoculate the explants onto the culture medium under sterile conditions to initiate the cultivation process. Control of cultivation conditions requires temperature and light, humidity and ventilation, as well as the cultivation cycle management. Adjusting temperature and light conditions according to plant growth requirements promotes cell division and differentiation. Maintaining appropriate humidity and good ventilation conditions helps prevent mold growth. Regular observation of growth conditions allows timely adjustments to cultivation conditions or media replacement, benefiting vegetable growth. Next, we will explore the application of tissue culture technology in vegetable seedling production. Rapid propagation technology includes 3 main aspects. First is the establishment of asexual propagation systems. Taking specific vegetable varieties, such as potatoes, as examples, these crops are often propagated through rapid propagation techniques. Second is microstem tip culture, which utilizes tissue culture technology to rapidly propagate superior varieties and enhance propagation efficiency. The last one is the analysis of application examples. Through microstem tip culture technology, virus elimination is achieved, resulting in healthy seedlings. The content on virus detoxification and testing includes the principles of detoxification techniques, methods for virus detection, and evaluation of detoxification effectiveness. Tissue culture techniques can remove viruses from the plant body, thereby restoring plant health. In methods for virus testing, techniques such as enzyme-linked immunosorbent assay and polymerase chain reaction are widely used in tissue culture. Additionally, for plants that have undergone detoxification treatment, the evaluation of growth conditions and virus levels is necessary. Genetic improvement and new variety cultivation involve multiple technical aspects, including genetic engineering techniques, maiden selection and application, and molecular marker-assisted breeding. By combining gene editing technology, we can improve specific traits in vegetable varieties. At the same time, using tissue culture technology helps to identify beneficial mutants for the cultivation of new varieties. Furthermore, the application of molecular marker technology aids in enhancing breeding efficiency when selecting for desirable traits. Finally, let's discuss operational examples of tissue culture technology and case studies. Taking potato detoxification cultivation as an example, virus-free potato seed tubers can significantly increase potato yields, generally by 30% to 50%, and can even reach 115.7%. Virus-free potatoes reduce the incidence of diseases by eliminating viruses from within. The seedling stage, budding stage, and flowering stage are all advanced, allowing the potato tubers to start swelling and growing earlier. The key technical links include selection of high-quality virus-free seed potatoes, processing and sprouting of seed potatoes, selection and preparation of soil, sowing and transplanting, field management, propagation and production of virus-free seedlings, as well as harvesting and storage. The steps for shoot tip detoxification begin with seed selection, followed by virus-free seedling to obtain the breeder seeds, which then develop into foundation seeds, followed by primary and secondary seeds, ultimately leading to the cultivation of the desired commercial tubers. The main steps in shoot tip detoxification include selection and treatment of materials, sterilization and disinfection, shoot tip excision, inoculation, cultivation, and virus testing. Thus, it is evident that tissue culture technology has been effectively applied in the breeding of superior potato varieties. In this lesson, we explored the key points of vegetable tissue culture seedling technology and discussed its applications and significance in modern agriculture. I hope that through this learning experience, you will recognize that innovations in agricultural technology are not only crucial for advancing agricultural development, but also an important means of ensuring national food security. As future agricultural scientists, we should inherit and promote the spirit of exploration and perseverance exhibited by scientists, applying what we have learned to practical work. After class, please complete the 2 exercises. Okay, that concludes today's lesson. Thank you all for listening.